Friends, welcome back to my channel. And today in this video, we are going to study about myeloid antigen presenting cells. In the last video, we learned about granulocytes under myeloid lineage cells. Today, again, we are dealing with the cells of myeloid lineage only, but they are categorized under antigen presenting cells. That means they present antigens to adaptive immunity. So, under antigen presenting cells, we have monocytes, macrophages, and dendritic cells. Now, these antigen presenting cells, they form cellular bridge between innate and adaptive immune system. Why? Because whenever any antigen enter our body, these cells will interact with this antigen, and after interaction with the antigen, they will present some part of the antigen to adaptive immunity, that is T cells or B cells. So they form a cellular bridge between innate and adaptive immune systems because they themselves are the cells of innate immunity and they mediate adaptive immunity by presenting the antigens to these T cells and B cells. These cells respond to pathogens and secrete some proteins that attract and activate other immune cells. They ingest pathogens via phagocytosis, digest pathogenic proteins into peptides, then present these peptide antigens on their membrane surface. As I told, they will interact with the antigen. After interacting with the antigen, they will present some part of the antigen to these T and the B cells. So some part means if this is the antigen, they will engulf the antigen and they will degrade it. After degrading, on their membrane, they will present some part of the antigens and they will go to the lymph node to present these antigens to the T lymphocytes. Then T lymphocyte, with the help of its adapter, will interact with this antigen. And that's how they to activate the adaptive immune system. These antigen presenting cells, they express a set of co-stimulatory molecules required for optimal activation of T lymphocytes. That means apart from presenting this antigen, which is necessary for the activation of T cells, but they can cause only partial activation of these T cells. For complete activation, they will express some set of co-stimulatory molecules as well. Now, dendritic cells presenting antigen to and activating the naive T cells. Macrophages and neutrophils, they form the first lines of defense against pathogen. Neutrophils, we already studied in the last videos. These neutrophils come under granulocytes. So, neutrophils is the first dominant cells of our body that interact with any antigens. So, coming to the first line of defense, these neutrophils and the macrophages from the antigen presenting cells, they constitute the first lines of defense against pathogens. Now, coming to monocytes, monocytes constitute 5 to 10% of white blood cells. They migrate into the tissue and differentiate into the macrophages and dendritic cells. Inflammatory monocytes enter tissue quickly in response to infections. Patrolling monocytes, a small group of cells that move slowly along blood vessels, provide a reservoir for tissue-resident monocyte in the absence of infection and may stop rather than initiate immune response. That means they migrate into the tissue and differentiate into the macrophages. That means macrophages are the differentiated form of these monocytes only. Inflammatory monocytes, they can enter into the tissue quickly and responds to the infection because they are already in the inflamed state. So basically these monocytes, they patrol, they are like a patrolling police in our body that helps to fight with the infections. And they differentiate into the macrophages, leading to enhanced activity, that is phagocytosis, and presenting the antigen to the cells of our lymphoid organs. Now coming to the macrophages. Macrophages means in the last slide only we learned they are the differentiated form of monocytes. So monocytes that migrate into the tissue in response to infection, they differentiate only into the macrophages. The important role of macrophages is they help in regulate tissue repair and regeneration. They participate in the innate immune response because these are the cells of innate immunity. Macrophages destroy pathogens by phagocytosis and present it to the T cells because these are antigen presenting cells. So what they do, they phagocyte, they digest, and some part of the antigen they present to the T cells. Osteoclast in bone marrow, microglial in central nervous system, and alveolar macrophages in the lungs. These are the example of tissue specific macrophages. Macrophages are found in all the tissues of our body, but specifically in bone, the macrophages are known as osteoclasts. In central nervous system, the macrophages are known as microglial cells. And in lung, it is known as alveolar macrophages. Inflammatory macrophages shows greater phagocytic activity and increased ability to kill ingested microbes, increased secretion of inflammatory and cytotoxic mediator, and ability to activate T cells. Now, how it functions? Macrophages, they will take up any antigen, engulf the antigen, they will digest it and present some part of the antigen to the Th cells. And that's how it helps in the activation of Th cell. Now, once the Th cell gets activated, this Th cell in turn again help in the activation of macrophages. So it is like a give and take. Macrophages is helping T cells for its activation. In return, Th cells are also helping macrophages for their activation. So macrophages and Th cell facilitates each other activation. Now, one more function of a macrophages is that they perform the process of opsonization. Opsonization means whenever any antigen, they can engulf antigen, but if this antigen is bound with any antibody, then this process of ingestion becomes more faster or effective, or the rate increases. The process of phagocytosis, this rate increases, and this is known as opsonization. Clear? So opsonization means they can engulf the antigen without binding with antibody also, but if antibody is bound with antigen, then the process becomes fast, and this process is known as opsonization. Opsonization means when the phagocytosis process increases, the rate increases because the pathogen is bound with antibody. So this is the method of opsonization performed by macrophage. Now coming to the dendritic cell, they are critical for the initiation of immune response. They acquired their name because they are covered with long membranous extension that resemble the dendrites of the nerve cells. If you remember, the nerve cells structure of nerve cell, it is like this. So the dendritic cells also, they look like the cells are look like this only. So we call it as a dendritic cells. They arise from both myeloid and lymphoid lineages of hematopoietic stem cell that we already saw during the video of hematopoietic stem cell. They capture antigen in one location and present antigen in the another location. Why? Because they present antigen to the Th cell. So they will bind antigen and they will move to the lymph node to present the antigen to the T cell. Okay. So outside lymph node, these immature dendritic cells, they monitor the body for the sign of invasion by the pathogens and antigen. And they process this antigen and present to the T cells after entering into the lymph nodes. So this is the process. Like dendritic cell, they wander in peripheral tissue. As soon as any antigen enter in the peripheral tissue, they will capture the antigen. They will engulf it. How they can engulf it? By the process of phagocytosis or receptor mediated endocytosis or pinocytosis. If the size of the antigen is large, we call the process it as a phagocytosis. If the size of the antigen is small, we call it as a pinocytosis. And when this engulfment happened after binding with a receptor, that means if this is a dendritic cell, here receptors are present, the antigen will first bind with the receptor and then along with the receptor, it will taken inside the cell. So that process is known as receptor mediated endocytosis. Okay, and then they will migrate to the lymphoid tissue to present the antigen to the Th cell. So that's why it is said like they'll capture the antigen at some other position and deliver at some other position to the T cell. So the next is erythroid cells. They form erythrocytes, which contain high concentration of hemoglobin and deliver oxygen to surrounding cells and tissue. Even the damaged red blood cells release signals in the form of free radicals that induce innate immune activity. Next is megakaryocytes. These are large myeloid cells that reside in bone marrow and give rise to thousands of platelets. And the function of platelets in our body is in the formation of blood clots. It helps in the formation of blood clots. So whenever we get any cut in our body, it helps to form blood clots so that the blood present inside our body, to prevent the loss of blood present inside our body. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.